Hello everyone. Thank you for joining us today to celebrate the special occasion as we are starting the third season of the BioBalkan Collab Initiative. On behalf of the organizing team, I would like to welcome all of you to this first block of lectures, which will focus on the amazing field of herpetology. Uh, Today, we will listen first to a lecture from Vukasin Bielica from the University of Belgrade. He will be telling us all about the intricacies of dice snakes and how they feign death, rather interestingly. Um, he has done research also on other model organisms, for example, pond turtles and also fire-bellied toads. But yes, in this lecture, we will focus specifically on dice snakes. Then also the for the second lecture of today, we will be able to listen to our first ever guest who's outside of the Balkan region. But we figured that it makes a lot of sense for this story specifically, and it would have been a shame to not make this happen. And still, Marilena Stamatiu, she is in the Mediterranean, which is close enough to us, anyways. Marina will tell us about conservation efforts. She will delve into a topic that we really love here at BioBalkan, which is citizen science. Um, Marilena is a PhD student at the Open University of Cyprus. And in the past, she's also done a lot of research on grass snakes in Cyprus, which are very similar to the dice snakes that we have here in the Balkans. Yeah, currently she is working on the impact of road networks in Cyprus. So that's also very important, like for conservation. Okay, thank you. Without further ado, I think we can switch on over to Vukashin, and he can start delivering his lecture.
Okay, let me just pull the slides up. Just a second. Yeah, so first, thank you, Eddie, for the wonderful introduction. And I would also like to thank, of course, the initiative for inviting me and allowing me to share this research. I hope, besides the catchy title, that the talk will be of interest to you, and I hope to have more and more questions at the end. So I will tell you a bit about my PhD project and this wonderful, amazing model species that it's dice snake, and it's quite interesting and enigmatic behavior called dead feigning. Now, for like a brief introduction, because I do have to tell how this all started. Yeah, it started all like 6 or 7 years ago when I was like first entering this field of science and of herpetology. And I first saw a dice snake on the island of Golemgrad. Of course, I did not pay much attention before, and since then it was like a healthy interest that turned into an unhealthy obsession. And after that, more and more, I started learning about this species, working with it, and eventually I devoted 5 years of my life to the. Yeah, someone microphone is like really annoying. Yeah, if you could please. Yeah, sorry for this, but I'm hearing myself 3 times. It's quite difficult to. Yeah, thank you. Yeah, I devoted 5 years of my life to it, and I feel like really in place to pull this talk because I've been recognized by Eddie himself as an Atrix Desalata expert. Now, enough about me. I think we can move on to the dice snake. Like I said, it's a wonderful species to work with. It never bites. It's quite easy to introduce students to it. And yeah, it has lots and lots of fun stuff associated with it, which makes it a good model system for like ecology studies and behavioral studies. One of those traits that you can see here is that it's color polymorphic. So in layman's terms, it occurs in different colors. So on this slide, you can actually see 3 different individuals of 3 different color morphs. The most common one is the top one. You see like a green snake with dark blotches. We call it the dots dice one. You have uniformly green snakes and uniformly black snakes. And a fun thing also is that it occurs, this color polymorphism like occurs more and more as you go further down south in its range. So basically, the fun thing is it has lots and lots of different phenotypic traits that you can measure, compare, experiment on, and see what happens. When we talk about its range, the range is huge. So going from Italy to China, all the way down to Egypt, which is again fun for lots of like genetic and molecular analysis. But in our case, and what brings to the story of dead feigning is in its huge range, the dice snake is facing off lots and lots of different predators. And because of that, you need to develop different types of anti-predator defense, and of course, tailor its defense to different types of predators that occur in its range. This inadvertently caused it to have like a really large defensive arsenal, and I put a video which I hope it will work. I think it will, yeah. So this is one of the reactions that they display. So they bluff, they puff up, they hiss, they imitate that they're like a viper and that it's going to bite you, but like I said, it never bites. It's a species that never bites. And the amount of different behaviors and displays that this species do is like huge. And I think an entire talk could be devoted just to naming what different stuff they do. But like for the purpose of this talk and this time format, I've put it in like 2 or 3 boxes. So if you imagine that you're a dice snake or any type of prey species that is being predated upon by a predator, you have several different phases that you have to go through before you are eventually, I hope not, but you eventually get eaten. So before capture by the predator, during capture, and after capture. And it's in the prey's interest to end this interaction as soon as possible, because the sooner you end it, the further away are you from death, right? And in the case of dice snake, I've listed some of the behaviors that they do. If you're interested in all the different stuff that they do and how certain phenotypic traits and other behaviors influence its behavior, I'll link some of the papers that we've published, and I do invite you to check them out if you're interested in behavior and like this kind of stuff. But I want to devote your attention to these 2 letters here, so DF and DF, which stands for dead feigning. Dice snakes display dead feigning exclusively upon physical contact, so when captured by the predator or being released by the predator, and they, like other species of the genus Natrix, they display it quite regularly. Now, dead feigning itself is quite an interesting concept, and it's widely present. Well, lots of animals feign death. It has lots of names like thanatosis, apparent death, playing dead, playing possum. The last playing possum, the phrase comes from the American or Virginia possum, which also frequently plays dead, but you can also see that there are some similar characteristics between all those animals. So it occurs in invertebrates and invertebrates as well. But again, the similar characteristics are the animal is laying on its back. If it has extremities, the extremities are up. They release foul-smelling liquids from the mouth, from the cloaca or anus, and it all works like simulating to the predator, hey, I'm dead, you can let me go. And I'm quite certain that you've also been given this advice, especially when facing off bears. So in America, there's like a rhyme that goes, if it's brown, lay down, if it's black, fight back, and if it's white, say goodnight. But the main idea is if you face a specific type of bear, the brown bear, you should lie down and pretend you're dead, and the bear will leave you alone. And of course, if you think about it, it's quite counterintuitive. So if something is attacking you and trying to eat you, why would you just play dead and, make the job easier? And there are several specific sets of circumstances which allow this behavior to work. Now, most predators avoid dead or sick prey. Simply, it's adaptive to just skip a potentially sick prey or a prey that's been dead for a while, avoid certain type of diseases. But of course, this may not work if the predator is desperate enough or hungry enough to try and eat a sick or dead individual. Now, most predators are attracted to movement. There are lots and lots of fun experiments done with foxes and I think ducks and chickens when it was ethical to do so. Now it's not. So basically, they were putting like a dead chicken and a live chicken with a fox, and of course, the fox would chase after the live chicken. Then they upped the game and they put 2 live chickens and 1 dead chicken, and the fox would kill 1 chicken and leave it be and chase the other chicken that's running around. So basically, if you move, you're going to attract a predator. If you just lay still and don't do nothing, you have a chance of the predator not noticing you or thinking that you're dead. Again, some predators cache their prey or even cache the prey for later consumption or for the consumption of their young, which also provides an opportunity to escape. So basically, you can imagine that if a predator thinks that you're dead, it will drop its attention or possibly leave dead prey in the nest, and then the prey can again come back to life and run away. Of course, there is stuff like gape limited predators. This just means that the predators that are limited by the size of their mouths and the prey that they can swallow. And for example, birds, most generalist bird predators, they swallow the prey whole. So if you're like a bigger snake, you play dead, perhaps it's quite difficult to handle you and you cannot be swallowed easily. And in all the commotion, you can again try and run away. And the final idea, why should it work? These are some of the ideas that I think they're most interesting. There is lots and lots of ideas on how dead feigning should actually work, is that a predator can be surprised. Just, a snake that it's violently struggling, hissing, bluffing, and then suddenly going limp and going like apparently dead. It may surprise the predator again, and that's how it works in natural populations. Now, a fun thing to discuss would definitely be the physiological and genetic basis of this behavior, which is also quite interesting, but I sadly do not have the time to do that now, but I do expect some questions later after the talk. Now, when we talk about dice snakes and why feigning death in this specific species is interesting, there are several reasons. Now, first of all, we have some knowledge that the stress hormones are actually lower in dead feigning individuals. We can approximately assume how, on a physiological basis, this behavior is expressed. We know that when they feign death, they go limp or they even coil a bit, but they have no visible breathing. And in my experience, they do not breathe during dead feigning. And they also enter a state of bradycardia, which means that their heart beats very, very slowly. Apart from that, they expel musk, like they have these cloacal glands that are really stinky, and they also expel feces when they feign death. So it all goes to like appropriately addressed like a dead snake. So musk, feces, no breathing, no heartbeats, and they have like a really gaped mouth, which you can see from the slide, a protruding tongue. They stop tongue flicking, so any kind of sensory mechanism is stopped. And of course, they have autohemorrhaging, or they expel blood from their mouths. Now, we wanted to know, are all these behaviors just randomly appearing, or are there any kind of specific factors that influence the occurrence or even the duration of this behavior? And that was a part, just like a smaller part of my PhD project. And then, yeah, we started like researching and doing science stuff. And the basic idea is how the dice snakes bring it all together. So is it just a case of sending the right signals to the predator? If you look at these two snakes, it's evident that the snake labeled as B has blood in its mouth, and it's quite a clear visual signal in comparison to the snake on the left, or snake A. So is it like a case of all these behaviors working together and addressing this like picture of a dead dice snake? Now, we processed lots and lots of snakes, over 1,000 snakes during my PhD project, and we worked all over Serbia, Albania, and North Macedonia. And we first found out that there are local adaptations in the expression of this behavior, which simply means that some populations are more prone to expressing this behavior and some are not. And one of the populations that's really good at this behavior, and the population that has really good actors or dice snakes that perform death feigning most commonly, is the population of Golengrad in North Macedonia. Now, why should this be the case? First of all, this is quite a small island of approximately 18 hectares that houses approximately 10,000 dice snakes. So that's about almost 600 snakes per 1 hectare, which is quite a lot. So the population is quite dense, and you can imagine that if lots and lots of snakes are basking communally, which is quite a common appearance in Golengrad, a predator that captures one and it feigns death, it has all these other snakes crawling around and it might get distracted. So That's one thing to keep in mind. The other thing is this island is home to lots and lots of bird species, most of which are generalist predators. So the kind of that we talked about, the gate-limited predators that have to swallow the prey whole or catch their prey for their young or for later consumption. But the main idea is they have one main predator type, avian predators that are like supposedly susceptible to the defense, such as dead feigning. And again, like I said, behaviors such as communal basking are common, even reproductive behaviors where you have these big balls of die snakes, 20 or 30 snakes rolling around the beaches mating. Again, it goes to the idea of feigning death can be a good defense mechanism in this case. And the final note is that die snakes on Golangrad suffer for a form of island gigantism. They're quite large compared to the mainland counterparts. So you can imagine a snake weighing almost 500 grams suddenly going limp. If you're a bird, that can be quite a challenge to swallow. Now we got some quite interesting results, but before that, I do have to just devote like 2 minutes of your time to the Golangrad. And yeah, this island is a wonderful field laboratory. My team and I've been working there for more than 5 years, but the team has been working there for 17 years on 3 different reptile species, on vipers, on die snakes, and on Hermann's tortoises or wood tortoises. And you have lots and lots of papers to choose from to read if you're interested in the fields of ecology, ecophysiology, behavior, conservation. So I've listed some of the papers that I think you should read. And yeah, do read them and see if you're interested. And of course, we're always welcoming of students, as Zedi himself can testify to, students from Albania, Macedonia, Greece, and of course Serbia. And you can, of course, write to us and see if you can perhaps organize a little field trip to the island. Now, the results we obtained by devising a set of 2 field tests. So all our experiments were performed directly in the field, and we wanted to emulate like a real life predator encounter. What this meant was like we search for snakes, we capture them, and we put them through 2 kinds of tests that are designed to simulate like a predator grabbing them and trying to eat them. Of course, we are conforming to lots and lots of ethical regulations, and I assure you no snake has been harmed during these experiments. Now, the first test was the capture test. It's quite intuitive. You find the snake, you capture it. We always capture snakes in one specific way, so with 3 fingers holding them at mid-body in the air, and we noted what behaviors occurred in the first 30 seconds after capture, with our intention focused mostly on masking and smearing. So the behaviors that are associated with dead feigning and making the snake unpalatable or stinky, smelly. Immediately after this capture test, we performed the post-capture test. So you imagine you're a predator, you grab the snake, and you now want to handle it so you can eat it. So we performed a series of standardized handlings that lasted for 30 seconds, which included pinching the snake for 10 seconds, then transferring the snake from hand to hand for 10 seconds, and finally lightly stretching the snake for 10 seconds. After these 30 seconds have passed, we put the snake on its back, we moved away, and we waited. We measured the occurrence of dead feigning, of autohemorrhaging, so blood from the mouth, and we timed the duration of these phases. Now I want to quickly put your attention on the figure that is on the slide. So basically, this is like a short illustrative form of dead feigning in die snakes. So we measured the occurrence of dead feigning, which is like the yellow box in the bottom, and we wanted to see how long it lasted and whether the occurrence of these behaviors before it, so masking, smearing, and autohemorrhaging, actually impacted the duration of any of these phases. If you imagine like a dead feigning snake, it has its mouth wide open, and it's really easy to measure the duration of dead feigning in die snakes because they close their mouths after they come back to life. So the living dead close their mouths, come back to life, then they're immobile for a bit. They're gauging whether to start moving or not start moving. This immobility phase lasts for a bit. Then they start moving, but really slowly. They tongue click a lot. They look around, and that's the decision phase. So this is the crucial phase before escape, because you can imagine that if you start doing all of this too soon and the predator is nearby, you can actually attract the predator's attention again. And if you stay for too long, then your risk of being exposed to heat, to dehydration, etc., etc. So you have to gauge the risks involved in actually making an escape attempt. So the first set of results was that smearing and masking were quite common. Almost 50% of snakes actually perform this behavior. And we also found out that the juvenile snakes or immature snakes, young snakes, are not quite good at this. And especially they're not good at dead feigning, and they're not especially good at auto-hemorrhaging, so expelling blood from the mouth. This goes in line with the general hypothesis that young snakes should avoid generally going out in the open and exploring such risky behaviors because of their small size. They can get easily eaten, and it just doesn't work. So we continued on with the results on the adult snakes, so older snakes. Dead feigning was also quite common, so one third of our snakes actually performed death feigning, which is quite a lot and quite a significant amount of snakes feigning death. That's why we like to call the Golemgrad the island of the dead, because as soon as you capture a snake, 1 in 3 snakes will actually feign death for you, so that's quite a lot. And from those snakes that actually feigned death, almost a third of them actually expelled blood from their mouths. So you can see that the occurrence of these behaviors in the population that's quite prone to death feigning is quite high. And now we get on to the most interesting results, and that is the duration of dead feigning. I know this might be quite confusing with the box plots and the dots, but basically these 3 box plots represent the duration of different the duration of dead feigning in different individuals, and we group them by the occurrence of different behaviors. Now, the box plot on the left, you can see that the median duration of dead feigning is closer to like 15 seconds, and those are the snakes that did not perform smearing or masking or any kind of bloodletting. They just feigned death. So they just opened their mouths and remained immobile. The red box plot are actually snakes that expelled blood from their mouth, and the orange box plot are the snakes that actually both had auto-hemorrhaging, so blood and feces and mask and everything. And you can see that the actual duration of dead feigning can be shorter in the snakes that actually performed all of these different behaviors. And this points on to the conclusion that these behaviors are working actually synergistically. They increase the intensity of the reaction, so these are better actors, and their performance can last shorter because they're more believable to the predator. And it's quite intuitive to explain. So smearing, you can imagine that if you smear musk and feces all over yourself, you will be it will act as a repellent to the predator. Perhaps you can actually smear the musk and feces on the predator as well. And sadly, we do not know, but perhaps there are some chemical properties of the secretions of the cloacal glands that work in conjunction with this hypothesis. The blood, it's not quite easy to explain, because we still do not know how actually is this behavior performed. In some lizards, they have this specific kind of muscles around their eye, which contract and they expel blood from their eye, which is been proven, but we do not know how dye snakes actually do that. We have some theories, and we are working on actually proving that these theories are correct, but sadly I do not have any info on how actually they perform this, but we can imagine that it's quite a clear visual signal, or perhaps an olfactory signal to the predator. Okay, there's blood, but this snake is definitely dead. This blood can actually be toxic in some species of snakes and lizards, so perhaps this is also the case with dye snakes. And it's not excluded that this blood actually contains some volatile chemicals that can be acute to conspecifics about the presence of the predator, and that also impacts the behavior as a whole. And before I summarize, I want to stress one thing out, and that is that we do not have enough natural history observations of these behaviors, and we are working in quite a specific set of circumstances, again with ethical regulations. And do keep in mind, we're not actual predators of snakes, so we lack enough context of how these behaviors are actually employed in real life situations with real life predators, and we can only assume how and theorize how dye snakes use them before we receive some kind of video material or observations in the field of are these behaviors actually effective. So to summarize, there are local adaptations in this behavior, which actually means that there are specific sets of circumstances that can make this behavior rather effective and in turn adaptive, and they can this behavior can actually increase the fitness of an individual if performed correctly and the right time in the right place. In the case of Golengrad, this is like a dense population, lots and lots of individuals, lots and lots of snakes, a specific type of predator, and a specific type of morphological adaptation, such as a large size, which makes them easier, harder to swallow, harder to handle. And of course, the case of the Golengrad ice snakes proves an ideal natural setting where such phenomenon can be studied. And yeah, again, I think I just briefly glanced over the entire behavior, and I would like to hear questions in the discussion because I think if I went on and devoted enough time to the ice snake, this would be like a 2 hour long presentation. So yeah, thank you for your attention, and I hope I was clear enough and that I piqued your interest into discussing these wonderful snakes and their behavior.
Thank you so much, Lukashin. Yeah, as I already mentioned, unfortunately, I am in the car right now, but I would just like to give the word now to Marilena. We'll continue with the second lecture of the day. Thank you. Yeah, and if anybody has any questions, please let them in. Please write them out in the chat so that we can read them at the end. Thank you.
Okay, hello everyone. I hope you can hear me. Let me share my screen. Okay, can you hear and see?
Yes, we can hear you and we can see you.
Great. So, hi everyone. I want to thank the organizers for inviting me for this talk, and I also want to thank all of you for spending your time here with us to talk about natrix snakes. So, as you might have seen, my presentation is on citizen science initiatives for herpetofauna, something that I'm quite passionate about. And while I'm not from the Balkans and I'm from Cyprus, which is a very Mediterranean, very small island, there are some things that we can share as experiences, and I hope you will have some questions and discussions after this presentation. So first things first, a small introduction. As mentioned, my name is Marilena Stamatiu. I'm from Greece and I've been working and studying at Cyprus for the past 8 years. I'm a biologist and an environmentalist from my bachelor's and master's. Um, I did part of my research on agama lizards, Stellagama stelio, or currently, because the taxonomy changed, Laudakia cypriaca, the Cyprus starred agama. And my master's thesis was on the Cyprus grass snake, natrix cypriaca, for which we will be talking a little more later today. I'm currently wrapping up the first year of my doctorate on road networks and fauna. So I'm a little farther out from herpetofauna, but it's still my passion and I really enjoy working on it. Some more clues as to how I'm spending the free time I do not have is that I'm a research associate at the Open University of Cyprus. I'm a member of the Herpetological Society of Cyprus, and I'm one of the founders and the maintainer of the Cyprus Herp Atlas. And I also volunteer in various other organizations. So this is the boring first part of this presentation. I'm not sure how many of you are familiar with citizen science as a concept, although I'm very sure that most of you have partaken in citizen science in one way or another. But just to set a baseline for this presentation, the official definition per the Oxford English Dictionary is that citizen science is some kind of scientific work undertaken by members of the public collaborating with professionals or scientific institutions. So, to put it simply, it's just a very vague term describing citizen and public involvement in scientific work in some way. So, while many of us, me included, for a long while thought that citizen science is something quite new because currently we have mobile phones, we can record videos, we can take photos, we can record and send data very easily. Apparently, it has existed for a very long while, and like many other things, bird watchers did it first. So the first examples of citizen science come from the United States from 1880, where an ornithologist asked members of the public to send him notes on bird sightings. And this is one of those notes on passenger pigeons. So that was the first citizen science initiative we have. And the second one was also from bird watchers on the United Kingdom. And herpetological initiatives followed around the mid 20th century. So why do we like citizen science? Why do we need to integrate citizen science into, let's say, pure scientific science done by a scientist? First of all, it has benefits all around. Citizen science is a great way to combine educating the public with doing research and data collection. You can show people practical things, how to collect data, how to come in contact with animals and nature. And at the same time, you can kindly ask them to help you and work for you and your data collection. And for this, citizen science initiatives are great for long-term and wide coverage geographically projects because as a scientist or as a small team of scientists, your funds, your finances, and your time are quite limited. You might have a single year, 2 years to do a specific research, but both your money and your time will run out. But if you ask people to monitor and send you data about their gardens, about their local parks, about things that are close around them, you can collect a lot of information for very limited, in a very limited or almost non-existent budget. So, if handled correctly, citizen science is a way to very efficiently collect very big amounts of data. And also, usually this data is also publicly available, which is always great for both scientists and the public members that want to have access and take a peek. And also something that I will discuss later on is that citizen science can use existing knowledge. Someone, say a farmer, for example, might have a species in their garden, but they don't think it has any value. And there is a scientist out there looking for this species. So citizen science is a way to connect these two people and actually make use of this data from one end to another. So what does this imply for a scientist? Do we just sit in our offices and wait for people to send us data while doing nothing? Well, of course, that's not how this will work. And I don't think any of us want this to work like that. But it means that our role as scientists, when we involve ourselves into a citizen science initiative, will change. You are not just a field scientist anymore. You have to educate people to show them how to collect the data for you, to show them how to hike in the nature safely, how to make a first aid kit, how to handle animals if needed. You have to guide them step by step, not only for their safety, but also to ensure that the data they will be sending back to you will be of good quality. So it is a very big investment in time, at least. And then you have to be an assessor. You have to assess the data that is being sent back to you. Are the people you have trained or overseeing good enough at the data they are sending? Can you use all of it? And what are the limitations to its use? And of course, for all of this to happen in this time and age, you have to be a social media manager. You have to be able to communicate and connect with the public effectively and make your cause, let's say, marketable in order to have people understand why you want them to help you and make them actually want to help you and have some benefits themselves. So I was thinking of how to handle this presentation because I really love grass snakes and I really love citizen science. And I think it's in Cyprus, we have a great way to connect these two things. So what I will attempt to do, starting from the less boring part of the presentation, is to tell you a story about the Cyprus grass snake through the lens of citizen science. So starting with some facts, we know that the Cyprus grass snake, Natrix cypriaca, which is pretty similar to all other grass snakes everywhere, although it does not play dead, it just masks a lot. We know that it has been a native species with established existence because of bones found in ancient establishments. But the first official record of it comes from 1865, named Tropidonotus natrix, which was basically the name for all grass snakes around Europe, as you can see in these very cool historical sketches. From 1865 to the mid-90s, there was not much research because research was done by hobbyists, by rich people who would travel and just take notes. But in the 1960s, due to very extensive use of chemicals in Cyprus and all around Europe, as you can see, there are some quite shocking pictures of DDMT use. So chemicals being sprayed by airplanes on ship, but they would also spray people, they would spray rivers, they would spray the forest. So because of the extended chemical use and also the urbanization, the creation of many new road networks, the Cyprus grass snake was thought to be extinct. And apparently nobody checked that. They just said it's extinct. But then in 1992, it was found again in a specific dam on the island. And that ignited a bit of research by very few people, and notably by Brigitte Blossat, who did her doctorate starting 1998, and she worked until 2008 on grass snake. And she basically did the only complete search on grass snakes we have until now. She calculated population dynamics, she mapped the known distribution, she made notes on the life circles, on pretty much everything she could do with her resources at the time. So we had some research going on until 2008 when Blossat left Cyprus to go to Germany, I think. And then after 2008, this research just stopped. There was nothing being done about Cyprus grass snakes. We were aware of their existence. There were some records, but there was no research, nothing. And that is until 2019. And that's where citizen science comes. So on the left side, you can see a map of Cyprus. And I'm not sure how visible they are on your screens, but we have these little red areas. These 4 areas are the Natura 2000 designated sites for the protection of the grass snake. Until 2019, we thought that the grass snake distribution is very limited only in these specific sites. And then 2 citizens go on a hike and they send reports of grass snakes from 2 areas outside these Natura 2000 sites. And this sparks a research. So I was part of this research. It was a very fun time. We basically would spend our whole day hiking around the mountain and looking for the grass snakes. And it did pay off because we found 13 specimens outside the known distribution, which greatly expanded this area of distribution for the snakes. And there is also a publication where you can see that we thank these 2 people for igniting this interest, because if they didn't report the snakes would still be there, but nobody would know about them. So in 2019, there was this spark to start working on grass snakes and find out what is the condition of their population. Are they really going extinct as we thought? Are they very limited? Or is there something going on but nobody knows about it? So we decided to start the investigation. One of the first steps we did based on that was the creation of Herp Atlas. The Herpetological Atlas of Cyprus, you can also access it in English, was created in 2022 after some initial work on the grass snake. And at first, we attempted to collect all the existing information from books, publications, and public reports. But then we realized that we can use citizen science. So we created a website, we created social media account, we started asking people to send us information, and we created basically a small encyclopedia that was easy for the public to access and use. And it also includes basic descriptions, maps, and photos of the animals. And it worked. In fact, currently we have more than 7,000 records of all herpetofauna species at Cyprus. We have 23 terrestrial herpetofauna species, and 3,000 of them are from citizen science records after 2022. Additionally, when we started, just by book and publication records, we only had about 50 records for the Cyprus grass snake. Currently, we have more than 2,000. I think yesterday I was looking at the database and we have, not 2,000, 200. 217, which is really great and exciting. And of course, it gives us a lot of information to continue research on distribution and other kinds of work. And then in 2023, we got a job thanks to a conservation grant by the European Herpetological Society to monitor the population of the Cyprus grass snake at Paralivni Lake, at one of the 2 places we can find populations on the island. And this is a nice area. You can see some photos. Some parts are very polluted, but there are also these lakes, these marsh-like areas that are great for grass snakes in theory. There is also a lot of food, as you can see. But the issue was that prior to the start of this research, I've only been to Paralivni Lake 2 or 3 times. I was not familiar with the area, so I could not really navigate and figure out where to start looking for the snakes. and you can see a satellite image of the lake from above. And it's not really a lake; it's a system of connected ponds with a canal in the middle. And at winter you have flooding and a lot of mud. At summer it dries up, so it's a pretty weird and difficult to navigate situation. So we knew that the snakes are there because many people would report grass snake sightings, but we didn't know where to go and find them. And finally, the first answers to push our research came from Facebook. So there is this group in Facebook called Biodiversity of Cyprus, and there are a lot of wildlife photographers there. And at some point, a colleague sent me a post of a grass snake from the lake and told me to go talk to the person that posted it to find the location. So we collected information from 2 or 3 wildlife photographers from the area, and in fact started getting some good pointers of where to look for the snakes. And we did in fact started finding the snakes. So that was not on purpose citizen science, but it was citizen science nonetheless. And lately, at the start of 2024, with the previous experience in mind, we got the Herb Trust project. It's an Erasmus Key Action 2 European project that has 2 main goals. First, to update the maps in our Herb Atlas site, and you can see here that we will have those interactive maps, hopefully in the next 2 or 3 weeks, that can also track your location and tell you what kind of herbs you can find in the area around you. And also to create educational material about herpetofauna, about influence of human activities on herpetofauna, and about citizen science and herpetofauna that we can use to further educate people and use on our presentations instead of constantly creating new presentations and new material. And all of this is publicly available, so if any of you are working on citizen science or herpetofauna, you can access and get ideas or even use the presentations if you want. And on this basis, we have the lessons uploaded. We did some excursions, some events. So it was a great time, a great way to connect with people, to go out in the nature, to have kids interact with lizards and realize that lizards and snakes are not as scary as they thought. And also, lastly, there is another project that was incredible. It's just wrapping up for creating a nesting site for Cyprus grass snakes. So we used all the data we had from citizen science, from previous research, to create haystacks for the grass snakes to nest, so we can hopefully find and count eggs. And it was incredible that the community came together and helped us by bringing materials, by bringing extra hands and equipment to work, and they were very interested and not as hostile as we thought they would be at the start. So, what is the recipe for a good citizen science initiative? First of all, to have a goal. What do we want to achieve? We don't just collect data for the sake of collecting data. Secondly, to have a structure. Who is the coordinator? Who receives the data? Who will the public message if they need more information? Then you need a lot of patience. You need to answer the same questions again and again. Sometimes you need to wait years until you have enough people to work effectively. For these reasons, you also have to create protocols to guide the volunteers to their work. You have to be open and communicative, and of course you need to have some media presence so you can communicate and inform all new members that might be interested. And by the way, these three photos are the three morphs, the three phenotypes of grass snake we have: the normal, the melanistic, and the picturata, similar to the dice snakes. And finally, is it worth it? Because you signed up to be a scientist and to do field work and collect data and all those cool things. And I'm here telling you that you should create social media pages and go talk to people instead of going out and catching snakes. Well, for many cases, it can be worth it, but it is a long time investment. You will need to be patient, and there is certainly a mental payoff from seeing people engaged and happy and willing to share this love for nature and these interests. So, if you made it this far, I want to thank you. And of course, I'm open to any questions you might have.
Thank you, Marilena and Lukashin, for this like wonderful lectures. Now we see like so many hearts. We have so many hearts that I get so confused. Like, I don't know, did anyone raise their hands? Or so now we have Q&A session where you can ask your questions.
There is one question for...
Yes, there is in messages.
I think that's for me.
Yes.
Yes.
Yeah, so it's like a two-part question, so I'll answer it in two parts. For the juveniles being less proficient and everything, not necessarily less proficient, but they adapt their behaviors, they correct their behaviors in the pre-capture, so they diminish their activity, they hide a lot, just so they don't have to get into risky situations such as displaying feigning death. Now we are working on it because in my experience, it would be apparent that the dice snakes have like a shy bold continuum where you have ones that are simply prone to death feigning and some that avoid it. And we have an experiment in place that is actually trying to confirm that the death feigning test and this behavior can be reflected in their personalities. So you can imagine like shy snakes and bold snakes in the context of feigning death and not feigning death. And regional variants, yeah, there is lots of regional variants where we showed that as you go further down south, there's regional variants in phenotypic traits and in turn in behavior, but these are mostly localized, so there's not like a cline of behavior that can be observed like in some insects where you have like latitudinal clines of dead feigning where simply from one longitudinal line and they just start dead feigning for whatever reason they do. This is not the case with dice snakes. And the last part, what would impact the juveniles to refine the behavior? Honestly, it's a fun question, but I don't think I have an answer for you. I think most of it comes from like they know to dead feign, or like when they are first born, they don't do anything, so they just exist, and approximately 1 or 2 weeks into their life, should I say, they start doing things like hissing, masking, and stuff. And I think we have not yet observed when exactly does that feigning arrive, but I think it would definitely do some with like some genetic basis of the expression of the behavior, and of course experience. And we know that they learn. Why? Because we use like injuries as proxies of previous experiences with the predator and snakes that are injured. they tend to avoid some of the risky displays. And one paper that I can link it in the chat, called the dicey situation. You will see that snakes that had injuries, I think they masked less, and I think they display this bluffing behavior more or less. So it would imply that they avoid this situation. So smearing is only and masking is only effective if predator tries, to smell you and not bite you. But if it bites you, you would have an impression of the predator, so an experience with the predator. And if you had a bad experience, you should avoid it. Yeah. So I hope that I answered the question. I think most of it. Yeah. No, thank you for the crunched.
So you mentioned a project where you build nests for collecting eggs. Can the same methodology be achieved with most snakes or just the ones in the question?
So, good question. Thank you. So, the methodology we used was first tried in the Netherlands by the organization Ravon. It's basically creating a hay stack with a stack with organic material like hay, manure, tree branches, and other stuff that the snakes will hopefully find attractive enough to nest in. So I am pretty sure this has not been tried for other species, although other species can and will use the stacks. Um, but it has been used for the genus Natrix in general. The only issue is that depending on your climate and the specific area, you have to set different humidity and temperature parameters. Because when they started in the Netherlands, they needed to create something warm for the snakes. While here at Cyprus, we just need to keep some humidity in the summer, but we do not need to keep the stacks warm because it's already very hot outside. Um, so I know it certainly works for grass snakes. I have not seen it being done for other species. In theory, it would attract other species, but probably not with that kind of accuracy.
Okay, thank you. So I have another one. Do you think that there is a need or possibility for EU funded life projects for some snake species?
As a general question for both of us, I guess.
No. It was more for you, Marilena, but if Ukashi wants to say something, he can.
I mean, certainly there are many possibilities for. You mentioned a life project.
Sorry, can you repeat?
You mentioned a life project or a horizon project.
Life projects, EU projects.
Mm-hmm. There is certainly the possibility to do a lot of things, but it takes a lot of time, a lot of coordination. So if there is something specific any of you want to discuss, we can exchange emails and talk about it. I am certainly open to the possibility of doing anything with snakes.
Vukashin, do you maybe have something to add?
Yeah, I'm also working with snakes that are like the meadow viper or Vipera orsini, especially in Macedonia, which, as you know, is extremely threatened. And I think there is a possibility, but I would have to agree with Marilena. It just takes a lot of dedication. And I think like the regional life projects, I think it's for the Mauremis in Croatia, which is doing really good. We have something similar with Rafford-funded projects in Serbia for the European pond turtle, and I don't see the many reasons why it should not work with snakes. But I think in the case of Natrix genus, I think most of it they are listed as non-threatened and stable populations, and I think more and more studies should be devoted eventually to reach a life project would be to establish population trends in a wider region. But I think the Balkans, and of course other regions such as Cyprus, as Marilena said, are like a black spot, the data deficient for some of a snake species. And I think, yeah, there's definitely the possibility. And of course, if any, yeah, I will also exchange my email with anyone who wants to talk a bit more and organize something. Yeah, so why not?
Okay, I have another question for you. Do you believe feigning behavior is an adaptation to lose less energy than some other defensive options? And is there a significant difference between natrix species in their feigning behavior?
Yeah, that's an excellent question. So we did not talk about the physiological implications of feigning death. And one rather good theory is that feigning death is like energy effective behavior. But the question that arises from death is ties directly into your other question, and that is that feigning death differs. Death feigning, when simply stated as death feigning, it encompasses a wide range of behaviors. And some call it, besides death feigning, apparent death, like I talked about, like tonic immobility, complete immobility, and there are lots and lots of different minute differences that can impact the energy effectiveness. So for example, the grass snake, at least in Serbia and Macedonia, they can feign death for like 45 minutes. And we actually measured one individual going like, I think 47 minutes, but it was like too hot and we had to move it just so it did not escape harm. So again, some other snakes the Zemenis longissimus, they also feign death, but they are quite tense, and they, I think, exhibit quite a lot of energy in keeping their body tense. So I don't know if that theory is correct. It can be in certain species, but again, you have to consider that feigning death, actually, you perhaps lose mating chances, you lose feeding chances, and it's quite an all-in behavior. So I myself would consider it that the hypothesis that it's an energy effective behavior can be true in specific cases, but I think in the general terms not. And I think most of the differences in other snake species playing dead is actually the postures and the being tense or not being tense. Again, for example, in die snakes, they're almost always limp without breathing and without visible heartbeats for a long time. And then you have like longissimus snakes that are quite tense, and you can actually see them moving and tensing and breathing. So there are lots of like specific differences. You just have to be in the field a lot and look at snakes a lot to spot them.
So there are two questions in the chat. Thank you, Nicola and Tanya. Nicola asked regarding CS, how does one go about implementing quality control during data sampling done by citizens or eliminating sample bias? For example, everyone loves to hike on this particular road, so most of the data is from that section and other regions become underreported.
So, yes, that is a very good and very important question for research. We have a lot of bias issues with citizen science. First of all, we have over and under reporting. For example, here at Cyprus, and I assume in many other places, we see over reporting close to the cities and under-reporting at more remote areas. So it doesn't mean that there are more snakes or more lizards close to the city. It means that there are more people to report these animals. So that's the first thing. And Also, we have over and under reporting based on species. There are many cases and quite well discussed in the literature where very common species end up being underreported. Because if you see a very common species of plant, for example, you will not make the effort to report it because you will think it has already been reported. And this is quite a complex situation. There is no real solution for all of these cases, but depending on your data and what you want to use the data for, first of all you have to report what kind of bias you have or you think you have and why this is happening. And then there are some ways to statistically eliminate the biases, but it's a case per case thing.
Okay, thank you, Marilena. So there is another question. What proved to be the best structure for a citizen science project? How was the work split up, and how many people does it take to run a citizen science project, in your opinion and experience? You mentioned marketing and communication with the public, data checkup, organizers, etc. So were there teams of people, or did one person do multiple types of work? Thanks in advance.
So I would say that the more the merrier for the organizers. In many cases, you have one person doing various types of work because simply there are not a lot of people knowledgeable enough or willing to help with that. But it largely depends on your resources. Certainly, it's very helpful to have people who are trained in communications, in marketing, in social media management, because it's one of the things that I find quite hard and tiring to do myself. But we are in a small country. We have a limited pool of researchers, so most of us are doing everything by ourselves. Um, for the other part, for the part of the volunteers, you don't really want large numbers. You want to have volunteers that are cooperative and willing to be guided and will bring you back good quality of data, even if they are not many in number.
Okay, thank you. And I want to ask, are there any more questions? So because there are no more questions, hi Eddie.
Hi, I'm on the bus now. Gonna go back to Skopje.
What an interesting life do you have?
You don't say. But yeah, I guess I would love to say thank you in the end. I'm sorry that I wasn't able to fully devote my attention to the lectures, but we do have a recording, so I can rewatch it later, and then we can have it on YouTube as well. We will make an effort to send an email with all the cool links, like the Herb Atlas. Like that thing is amazing. I've looked at it, and also the Herb Trust modules that I've only like scrapped the surface of. So yeah, and also some of Vukashin's links as well. We'll figure that out and send you a nice little email. Yeah. So okay, thank you everybody. I hope you had a good time like I did.
Thank you so much, everyone.
Yeah, thank you.
So Kashin and Marlena, thank you so much for these lectures. We will learn something. And I hope we meet by other chance. I believe that everybody enjoyed your lectures, and I hope that you and our other participants have a nice week. And that would be all from this week's lecture.
I think
Yeah,
you
thank you.
muted
Thank you
yourself.
for having us. Yeah.
All right. Thank you, everyone. Bye bye.
Bye.